For BIO106, and it's going to be some more background information about some of the things we're learning about this semester, and it's going to be about, a little bit about some add-ons about experimental design in molecular and cellular biology. And I'm a molecular biologist, and I ran into the topic in college, and molecular biology is the combination of genetics and biochemistry. So it emerged as a field in the 1940s and 1950s when scientists established that DNA was the basis for genetic inheritance, the primary basis for genetic inheritance in all cells and all organisms on Earth. So molecular biologists really like DNA. And when I told my high school friends that, when I was in college and I went back home, I said, I'm going to be a molecular biologist. And they're like, whoa, you're probably going to have to look at the electron microscope all day long. And the fact is that, nope, these things are too small to see even with an electron microscope. So no electron microscopy for me, and lots of light microscopy, but down at that molecular level, you can't see the stuff. And so experimental design is how scientists understand what's happening. And so this is going to be especially focused on the idea of experimental controls. And so, you can set up a experiment, and you have experimental samples. This is where we're doing our test. And then molecular biologists set up additional control samples to prove that everything's working as planned. And so here's a first example. What I'm going to do is I'm going to try to describe two examples. The first one is just totally made up, and it's not a molecular biology example, but it just helps you understand the concept of the controls. And then the second is a molecular biology example. And then for the homework, I'm going to ask you to invent an example where you think about doing an experiment. And your experiment could be, so it doesn't have to be molecular biology, right? That's not realistic. But you can think of an experiment that's more like this first example, where you can see what's going on, but you realize you need to have these controls that I'm going to describe. And hopefully, you can think of an example that's a little bit complicated, like this second example that includes the controls, to really help you understand what's going on. So here's the first example. The first example is focused on mice, and I like to give this example because it's one you can see and you can understand. So the experiment is, how long do a adult male and female mouse need to be together before the female gets pregnant? And so the experimental samples are just going to be, take these male and female mice, take a bunch of them, individuals, and put them together and separate them after different lengths of time. 1 minute, 10 minutes, 20 minutes, 2 days, like that. So a little time course to establish, we don't know how long it takes them to get to, each other and, have their species-specific courtship. So let's do this experiment, all right? And so it turns out you can buy lab mice, and you can buy virgin female mice. And so obviously, when they were juvenile mice, the company that sells them separated the boys from the girls, right? And so we have virgin female mice. And we're going to buy virgin females and virgin adult females and adult males. And we're going to put them together in an experiment. So we're going to order them. Here they come. And we need to do this, right? We need to just do a control where some of the females never get together with a male, because we need to prove as a control that these are virgin females. We cannot trust the company, right? We have to prove that, because if one of them was not a virgin, that would foul up our experiment, right? Like that, right? So that's called a negative control. And a negative control is when you expect to see no change. And sometimes there are a variety of negative controls. So in our second example, it's a little bit more complicated, and there are multiple negative controls to test each possibility to demonstrate the system is working as expected, even though it's too small to see what is happening. And when we set stuff up like that, even though the experiment is happening at that molecular level and the controls are happening at that molecular level, we end up setting up stuff that's like this, where you can see the mice, you see, understand that, yep, there are those mice, and you can see that, but we're not just going to watch them mate. right? Because we don't want to intervene. We also don't want to predict and expect that what we judge to be mating interaction is actually going to cause pregnancy. We want to know how long they need to get pregnant. So we're not going to watch them. We're just going to time them, right, like that. So but it's very common in molecular biology that you're doing stuff like we're doing in lab. Like we're working on the bench, and it's like we're working in a kitchen. Mixing stuff together, but we're actually testing things at a molecular level. And so I say that a lot of times this lab work is like clever kitchen work. You need to know the biology, you need to know what's going on to participate in the discussion of, what we're testing. But the actual planning and designing the experiment is got this kind of kitchen level work component to it, and also the kind of cause and effect logic. And what I've seen is that there are lots of students at USM who are smart enough to get this done really well. And so we've had students graduate with a bachelor's degree from USM and gone out to biotech companies and been literally stars, certainly good employees, because they know a key thing is knowing how to work hard and keep track of stuff. Like you're working in a kitchen, like, whose order is that, right? And you're like, well, I know, because I've been paying attention. Even if it's not my tables, I've been paying attention, like that. So first example, negative control. The females are stay apart to prove they're virgin females, like that, right? Then the next one is we're going to put a male and female where we're going to put a cage where there's a male and female together, and we're going to put a few examples of that, and we're going to leave them together, and they're going to be what's called the positive control. We're just leaving them together. That female is going to get pregnant eventually, and we're not testing it. It's not an experimental sample. We never separate them. We just leave them together. And that's called a positive control. And the positive control is where you are guaranteed to see the change. You will see the change, right? And so if you think about it, that establishes kind of the two extremes. The negative control is going to be no change. And if there's a change, there's a problem. And we can't just bluff our way through or fake our way through. We just dump the whole experiment. If we have some virgin females that have been separated as our negative control, and any one of those gets pregnant, we throw out the entire experiment. We just disregard it, And then similarly, positive control. Those females have to get positive, because here's a positive control possibility. Maybe the company had sold them, they're virgin females, but they're not reproductively mature yet. They're still juveniles, and they're not capable of getting pregnant, right? So we want to see all those females getting pregnant. And notice I said we didn't just set up one cage with one male and one female. We have to set up a bunch, and we have to set up a bunch of positive controls and a bunch of negative controls to get that data to be like, oh no, we had 20 positive controls and all those females got pregnant. We had 20 negative controls and none of those females got pregnant. And that, you see, that really sets up confidence in the experiment, like that, right? Okay, like that. Now, it's kind of interesting to point out that with our spectrophotometers we've been using, we actually are, when we calibrate the instrument, right, and we move the needle to here and then we move to there like that, we're actually doing that as a kind of equivalent to a positive and negative control, right? At first, to calibrate the spectrophotometer, the lights are out, there's no tube in it, it's completely dark. That would be equivalent to infinite absorbance, and that's like a positive control, that's what's going to happen. And that's the extreme, change version, or the extreme result. And then we take a tube, we put it in, a lot of times with water or some other zeroing liquid, and we set the instrument to zero, and that would be like a negative control where the light is just shining through it. And people wouldn't refer to the calibration of the spectrophotometer as a positive and negative control, but it's equivalent to what we're talking about, where we're setting the two extreme possibilities. None of the females get pregnant, or all of them get pregnant. So we set up a bunch of cages that have nothing to do with our experimental result or a hypothesis. They have everything to do with experimental design and confidence that our experiment is working, even if we're not watching the mice, not videotaping the mice, watching them every moment or anything like that. We can just confidently, leave and have them, set together for different lengths of time. Okay, and so That's that first example, okay? And so now we're going to go on to this example. And this is what we're going to look at. And it's a Petri dish. And so that's just a little sterile dish, and it's got some agar in it. And those are bacteria colonies growing on it. You can see some are blue and some are white. And so this is going to be a more detailed example. And it's a molecular example. And you'll see we have to set up multiple plates, not multiple cages, but multiple plates with bacteria to show that we're confident about what's happening. Okay, so let me step through that. So the first thing is that many of you have not really heard about a bacterial plate before. But here's an example of one where a scientist has just taken some material from nature and he's just spread it out on a bacterial plate. And so let me just show you that bacterial plate looks kind of like this. It has a bottom and a top, and it's covered and sterile. And then inside the dish, there is material that's gel-like, kind of like Jell-O. It's called agar. The agar is made up of a long chain of sugars, a polysaccharide that's isolated from certain species of seaweed. And when that agar is heated, it melts, and the liquid is it's a liquid, and then when it cools, it hardens into a gel-like material. So we have a gel-like material, and it's moist. It's got a lot of moisture in it, and it also has nutrients. And when the agar is hot, we get to choose what do we add for nutrients, right? And there are a variety of different nutrients to grow different kinds of bacteria or other microcellular organisms like yeast and fungus and stuff like that. And the organism is going to grow on the surface. And here's the key concept of a bacterial plate. If we look at it like this, is that if a bacteria lands here, it has access to those nutrients. It doesn't dive into the gel. It stays in that place because the gel is a kind of a stiff matrix, but the nutrients can diffuse up. There are no puddles of water, no puddles of liquid on the surface. So in a sense, it's moist and dry. And because of that, the bacteria cannot move around. So some bacteria have a flagella and can swim. Other bacteria don't have a flagella, but if they're in liquid, they just float around, they just diffuse around. And so what we've done here is we've trapped the bacteria so it's immobile, but it can still live, take in these nutrients, and grow. And what it does is it starts out as a single bacteria, and it divides, more, and we can get hundreds of thousands and millions of bacteria here, and they're all really identical twins. They all came from the same original bacterium that was there. And that's a key concept. It grows so much it becomes visible, typically overnight, over a day or two like that, it becomes visible, and that's called a colony. So if we go back to this image now, you can see what's going on here is these were just bacteria in nature, and it looks like, whoa, that doorknob was really contaminated. Well, actually, no, it wasn't, because there's just 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, like that. There were only about 50 bacteria that were originally on that doorknob, or whatever that source was. There really weren't that many bacteria. They've just gotten to land in that spot and grow so much that you see them. And microbiologists have really relied on that over the years, because when you can see the colony just by the way the colony looks, the color and the texture and so forth, and here's an image of that, the shapes of the texture and shape of how it's on the surface of the agar, that helps microbiologists identify what species of bacteria or fungus is there. And so this is just our first example, and the cells are spread out, and that's going to be the story here. The cells are spread out on the surface, and so there are visible colonies here. This was grown for a day in an incubator at the ideal temperature, and how many bacteria were there when the plate was first prepared and bacteria were spread on it, and the answer is looks like there's about 600, 700, 800, something like that, several hundred bacteria. We could try to count them, but yeah, and some are blue and some are white, okay? So there's that, and then here's the other major kind of plate that you'll see, and here we didn't spread the bacteria out smoothly. What we did is we spread them out in a streak plate, and that's called a streak plate, where we're actually just scratching the surface with bacteria. And so there are really two ways to prepare those plates, and either a streak plate or a plate like this. and it's how we add it. So here are these bacterial loops. They could be disposable plastic ones or sterilizable metal ones. And what that loop does is it picks up a tiny drop of liquid with the bacteria in it, and we can use that to make a streak plate. And we start here, and there were a lot of bacteria, and they started rubbing off the loop until there are very few. And usually what scientists are doing is they're trying to spread it out so they can get these individual colonies like that. Sometimes this is all one species. You can see the colonies all look really similar. Sometimes there's a mixture of species, and you can spread them out. So for example, they could have taken this and they could have done a streak plate, and initially there might have been a lot and they're all crowded together, but then we spread it out and we get these individual colonies and can isolate a specific one that we're interested in studying. So a streak plate with a loop, and then these are called hockey sticks, and that's how you spread the material out across the plate. So this plate was prepared with a hockey stick, and this plate was prepared with hockey stick. You spread those bacteria out, and again, or fungus, microorganisms out, and they grow all across this plate. And so different types. Okay, so now we're going to start talking about what's going on here. What's the experiment that's being set up here? And the bottom line is the scientists are trying to get the bacteria to make a gene from another species. For example, maybe they wanted to get this bacteria to make the human insulin. And just as a side note, when I was a kid growing up, diabetes was a very, very difficult disease because anybody who had diabetes that needed to get injections of insulin actually got injections of insulin using pig insulin. And they got the pig insulin from slaughterhouses where they would get the pig blood and they'd purify that pig insulin. That was the source. And the problem with it is that the human diabetic patient would start to become sensitive, almost like you're rejecting like a organ transplant, like that, rejecting tissue from a foreign organism. And so the person would start getting sensitive to it. So now anybody who gets anybody who has diabetes and takes injections of insulin, they get human insulin. And how did that work? How that work is, there's a lot of work, and it's not as simple as this, but the scientists took the human insulin gene, inserted into bacteria, and the bacteria grow huge amounts of human insulin because all it takes is the human insulin gene. It's more complicated than that because the story of the human insulin being fully ready as a protein takes steps that the bacteria can't do. So it's more complicated than that, but every person that it takes insulin now, it takes it's human insulin, and it was made in a vat by bacteria, okay? So this is how they did that. So they start out with this, and this is just a natural bacteria, so it's a bacterial cell, prokaryotic cell, and the prokaryotic cell typically have one chromosome, and so this is DNA, and this is the bacteria's chromosome, and it's really typically a circle. There's a lot of variety in bacteria on our planet, and so some are not circles and some are not a single one, but we're just going to use this because the most common bacteria that scientists use for this genetic engineering has just one big loop. And then, in addition to that chromosomal DNA, this bacteria happens to have some extra chromosomal DNA, and there are these small circles called plasmids. So that's plasmid, it's DNA, it's the same story, it's just a short piece. And it turns out it's pretty easy to gently break open, sort of poke holes in this bacteria so that the plasmid DNA leaks out and the big chromosomal DNA gets tangled up with the debris of the cell. And we can isolate or purify these plasmids, and that's called the plasmid DNA purification. And these are natural DNAs, and they're very common in bacteria. They're different, different forms of them and stuff like that. One thing that's pretty common is that you've heard undoubtedly that some bacteria become resistant to antibiotics, and lots of times those antibiotic resistant genes are actually on a plasmid. And that's problematic because they can share that plasmid with other bacteria, even sometimes other bacterial species, okay? So it can be something that's valuable for the bacteria, very helpful for the bacteria, and we can isolate it and work with it in a test tube. And in a test tube, it's purified plasmid DNA, just like that. Very kind of pretty easy to purify it and work with it in a test tube, and that's where we're gonna insert the human insulin gene, like that, okay? So once we have done that, then we can put this back into a bacteria and get the plasmids back into a new bacteria. The old bacteria are dead, and we have a new one. And notice that this bacteria doesn't have any plasmids, and we're going to add them in, right? And let's say that this plasmid has that antibiotic resistance, and this bacteria is currently sensitive to that antibiotic. And the antibiotic that we're going to talk about that's commonly used in these kind of experiments is called ampicillin. And ampicillin is related to the old-time penicillin. And ampicillin is an antibiotic that works great, but it's really never used in medicine anymore because there are so many bacteria that have become resistant to it. But in a lab setting, we can start with bacteria that are sensitive to the antibiotic, and we can put the modified plasmid in them, and now we've got the modified plasmid in that bacteria. And maybe again, it's got the human insulin gene. There are thousands and thousands of examples of what we might want to put in and study in a variety, just so many different ways that this is very, very common procedure. And getting the DNA back in is called bacterial transformation. And so we can do that method and get the bacteria in, okay? So then this doesn't work very well. It's not a problem with the science, it's just unnatural. The bacteria really don't want to take up DNA from the environment. It can occur, but it's very rare, so it's very inefficient. Maybe only 1 in a million bacteria will pick it up. So we need to be aware of that and have controls in our experiment to deal with that, okay? So that's just some background. Here's a look at that. So here's the plasmid DNA that would be in a test tube, and we're going to take this gene, and again, it might be the human insulin gene or any of thousands and thousands of other genes we might be studying, and we can insert it into the DNA, and we call that a recombinant plasmid because we've recombined the plasmid DNA, the natural bacterial DNA, with really often a foreign gene. And here's an example of a plasmid that has been modified by a company, and the company sells it because they've kind of made it, improved it for these experimental uses, right? It was a natural molecule out in nature, they isolated, and then they modified it. Here's the antibiotic resistant gene, and here's an origin of replication, so the DNA will get copied. And then here's going to be what we're going to be interested in, and this is the place where we're going to insert the foreign DNA. And this company did a clever thing. They have the insertion site where we're going to put our DNA in, again, human insulin. They put that in the middle of an existing bacterial gene. And so if we insert something, it erupts and destroys the bacterial gene. And they made this color blue because if this is intact, the bacteria will be blue. And that means there's no insert. Okay? But if we get an insert, this foreign gene gets inserted, it will interrupt that, and the bacteria will not be blue, they'll be white. And so that's what's going on here in that experiment. They're just using this as a way to see that the blue have a plasmid because they're on this plate, and this plate has ampicillin. Initially, they spread billions of bacterial cells on it, and the ampicillin killed all of them except the hundreds that happened, the rare event, they happened to get a plasmid. And we can see that some of the plasmids are blue. We're not interested in them. They were able to grow because they got a plasmid. They have that antibiotic resistant gene, but they're not what we want. We want the white ones because they're the ones that have the plasmid. They're growing on ampicillin, and they're not glowing blue, so they must have gotten insert in there. And so we want to pick the white ones. That's our experiment, like that, okay? So that's that. Here's just a picture of an example of a product that a company sells for different purposes. And it's not that blue-white selection scheme. It's used for a different purpose. And so these have been around since about 1980, so that's like 45 years, and companies design them and improve them for different applications. So it's very common, okay, to do this work. Here's just a little blurb on the blue-white colony assay. There's what we're looking at. And now we're just going to talk through this. This is going to be an experimental plate. That means we could get what we want off that plate. And it would be the white colonies. And there are a bunch of them. We probably only need that one. So we can pick that one. We can make a streak plate like that, save it. We've got it isolated. And maybe we pick several and study them all, right? Okay, like that, okay? But to set up this bacterial transformation experiment that got this on the plate like that, the scientists set up controls. And here are the controls they set up. So the first one is that they wanted to show that the bacteria are the bacteria sensitive, the bacteria they're using, are they sensitive to ampicillin? So one plate, a control plate they set up, is that they just spread the bacteria out on a plate that has nutrients, but it has no ampicillin. And the prediction, the expectation, the requirement is billions of bacteria grow. All of them grow. So that's a control, and it's kind of positive control showing, yep, our nutrients are in there. We're using a bacteria that's growing, that's living and healthy, and we're using billions of them, right? So that's our first control. And our second control is this. Same type of thing. We're not talking about the plasmid yet, but a plate with AMP and no bacteria, no colonies, none. If there's one, we're worried. And that goes back to those, positive and negative control. So that's a positive and negative control just on the starting bacteria. Are the bacteria what we want? Are the nutrients what we want? Does the ampicillin work? Does it work to kill all these bacteria? And we don't have any exceptions like that. Something that could happen is there could be a contaminating species of bacteria, maybe in our lab or something like that. And we get we get some colonies, and we're like, we got to get this lab cleaned up, like that. So those are our first two controls. Then our next control is this, where we want to just take what we would call uncut plasmid DNA. So remember, in our test tube, it's pretty easy to purify plasmid DNA, and then what we need to do is cut it and insert our gene of interest. So these are just our uncut starting material. Are they good? Is it the right amount? So let's add uncut plasmid, and we'll add it to a plate that has ampicillin, and we should see many, many, many bacterial colonies. So this is a control to show, yep, we got our plasmid, and yep, the ampicillin gene is working fine, and yep, we're using a lot of it, because this is our maximum. The uncut will be maximum. This experimental sample will be fewer, because we've caught it, we've glued it back together, it's not going to be as good as the uncut. So what's the most we could do? And let's make sure we start with that much DNA, that we get lots and lots and lots of colonies like that. So that's our third control, and that's testing the plasmid. And lots of different features of it, right? Do we have enough? Do we have the right amount, not too much, the enough amount? And is the ampicillin working? Ampicillin resistant gene working like that, right? And to rescue or cause these to live, right? Because in our second control plate we said, can the bacteria can they all be killed? So they're sensitive, and then can they all be rescued with the plasmid? Is that all working like that? So that's our third control, and I'm going to try to keep track of these numbers, but don't worry if I get them out of the number, get kind of messed up on the number. Then our fourth one is this. Our fourth control is we use the cut plasmid. So it turns out when the plasmid is cut, it does not go into the cell well. And so here, if it's cut well, we'll see very few, or hopefully zero plasmids. Zero colonies, okay? It just, because when it's cut and it gets put in, the bacteria just destroys it. It chews it up from the ends. So the bacterial plasmid or the bacterial chromosomal DNA, they really need to be circular because the bacteria has enzymes that will destroy them if they're cut like that. And that's a kind of defense mechanism against viruses, because a lot of times viruses are not circular and the bacteria can just destroy them upon infection, because bacteria can get infected with some certain kind of viruses. Okay, so this is cut plasmid. Okay, and just to mention this is going to be cut plasmid, and remember, we're going to glue it back together. And when we glue it back together, we don't want this to just glue back together. We want to glue it back together only if it has an insert. It only has that foreign DNA, that human insert delay. So when we cut it, it turns out we can cut it with, I'm going to make these different sizes, because there are enzymes that cut the DNA, and they cut it at different sites. So this is enzyme A, and this is enzyme B. And they'll cut, and we'll get rid of this piece, the original piece of the bacteria, and that will just kind of float away. And these won't stick together. We can't glue those back together. So we're calling this a double digestion. We cut with two enzymes. Okay? And so that's our fourth control, right? Okay, like that. And then here's our fifth control, and here, our fifth control is going to be that we have that double digestion, but we also have a control where we just cut with single digestion, just cut with one of those. And that would also give no colonies, so we could do that as a control, no colonies. And then our sixth plate is where we take that single cut stuff and we add the glue. And the glue happens to be called DNA ligase, and you don't need to remember all these names because you're not going to be quizzed on them. And the DNA ligase is the glue that can close it up. And so we do an incubation with that, and it should be able to close that up. And then when we do that control, we should get lots of colonies. And there, we're using this as a control to prove that our enzyme's working. So single digestion, not closed. It's cut, so no colonies. Then is next control. If it was incubated with the enzyme glue, it goes back together. We get circular DNA, lots of colonies. So we're proving that our enzymes work like that. Okay, and and so on to the next control. And we don't have to really test both of the enzymes. We could, but it would probably not be necessary. Just cutting, one was probably going to be enough. So the next one is that we do this, where we add, we cut with the double digestion. We use that material, and I'll just make big and little scissors to kind of designate the A and B. And then we take our insert, this is the foreign DNA, and we're going to insert that in to make the recombinant molecule. And we prepared that by also cutting it with the big and the little. Like that. And we add ligase. Okay? And this is our seventh plate, and this is our only experimental plate. The first six plates were controls. And so this is a good, little bit complicated example of experimental design in molecular biology. We can't see these things. They're far too small, even with the most powerful electron microscope. You can't see that. And so we got to have these controls. And so we have to set up lots of plates. And in some cases, there are many more plates, just to prove all the different things are working as we expect or as we need, like that, right? The system is working, and all the individual pieces of the system are working. Another way to think about it is when experiment fails and we don't get what we want, we have to troubleshoot it. We have to solve it. And controls are a way of helping to solve problems because they're really common. And so you set the experiment up, and you're like, oh, our controls are not working. We got to we got to get our controls to working. It seems like the ligase is no good, like that. And that's a product you got to buy, so you got to test it out, like that, And so this is actually an experimental plate, and this takes us back to that. So this is our plate number 7, and it's our experimental plate. And we see our blue-white colonies because they could have the insert, the colony insert like that. And In fact, you could say, oh, shouldn't we do another control? Because you can think and you can add more controls. For example, we should have, and we kind of did. I don't remember what the number was, but we could have added the chemical that is in the agar plate that causes the colonies to turn blue, to confirm, right, that our blue-white assay is working. And it would have been actually a good control to have, that in that earlier control where we're like working with the uncut DNA, right? And we're just seeing lots and lots of colonies, right? That they're all blue, right? And I should have thought of that and included that. But you can see that that's an important control also to show, yeah, we can get blue colonies, right? And they're all blue. We don't have some weird thing where even our regular plasma we start right already has some blue and white. So, we get very confident that those have that human insulin gene insert. But again, in the experimental plate, that's the rest of the plates just go into the trash. We write down the data, like that, and numbers of what we saw, and they just go into the trash, right? And so that's where we're at on that. Okay, so I want to just try those two examples, and now as the homework, what you're going to do is just take your time, think about it. It's really a thought question, but think of designing an experiment. It's not going to be a molecular one. It's going to be more like the mice, and it might be something out in the environment or something like that. So it's not going to be a molecular experiment, but think of an experiment that would require some controls. So something like the mouse example. Remember, the mouse example just had 2 controls, and the molecular example required a few. So just take time to think about it, because that is experimental design, and you can practice some of it. Think about it, and then describe your experiment. In the homework for this is just, invent an experiment, describe it, and explain what the experimental samples would be and what controls would be that you'd need. And that's it. That's the part that we're going to get out of experimental design. Okay? So you're all set with that. Thanks.